Presentation tonight, I'm going to give a brief background on my research. Then I'll talk about my species of interest, which is the ring Boghaunter, and I'll move on to what I did and what I found from the research. At the end of the talk, my aim is to not just share my findings with this broader community. I also want to get feedback and ideas. So, okay. So, there's been consistent loss of biodiversity on the Earth's surface, which has affected many ecosystem services and functions provided by these species. Now, according to Worldwide Fund for Nature, there is 52 percent overall decline in wildlife population, of which 76% is freshwater species. And then my species of interest, which is the ringbow hunter, happens to fall under this category. Um, However, according to Log et al., there is still a possibility of reversing this species decline. And according to them, by 2050, we should be able to attain the total or full recovery of this species or prevent them from declining through series of conservation efforts and then strategies. So in with my diagram here, we are in 2026, so it's somewhere here, and there's still declining of species. However, according to Locke, they said by 2050, through series of conservation efforts and strategies, we should be able to attain a total of full recovery of these species. Now, there are several conservation efforts and strategies that scientists and researchers use to address this issue of population decline or species decline. However, I'm just going to focus on one major conservation efforts called the monitoring. So, this monitoring is a strategy which involves you regularly observing and then recording information or data about a species, its population, its habitat, and then it will help you understand the trends that is occurring or going on within this species in their area. Um, and can also help inform other conservation efforts regarding the species of interest. There are different ways of monitoring species, and I'm just going to limit myself to three of these ways. The first one I'll talk about is the drone for area survey. So this is where you fly a drone over an area. It captures whatever that is going on within the species in that area and visually gives you data or information about the species, and it's very useful for population estimates. And there is also camera trap where just like the drone, you also set a camera in the area of interest. It captures whatever that is going on within that area and visually gives you data about the trends of the species or the population of interest in that area. Then there is also tagging, where you attach a unique identifier to the species of interest just to track the species. And this is very helpful in obtaining of information about the behavior, the movement, and also some population dynamics about the species of interest. So all these three are ways of monitoring species, and I'll call them traditional ways of monitoring species. Just like the saying goes, there's nothing perfect. So there are challenges associated with this traditional way of monitoring species. And again, I'm also going to limit myself to just three of these challenges associated with the traditional way of monitoring species. The first one is the decline in taxonomic expertise, which greatly affects the ability to identify and also classify species, which often leads to ineffective conservation plans and strategies. So when we look at this graph that I have over here, between 1970 and then 2020, there's been consistent decline in taxonomic expertise. And if there is decline in taxonomic expertise, we cannot confidently or accurately identify, classify, and distinguish between species. So that's one major problem that is associated with the traditional way of monitoring, since it involves recording, observing the species in the area of and then the second issue that I'll talk about is unable to identify and then differentiate between cryptid species. So when you look at this diagram I have over here, one might think these are the same species or these are the same organism. However, genetically, we can prove that these are different species. They could be of different population. Um, and so we are not able to identify and differentiate between cryptid species through just observing and then recording or visually looking at the organisms. And there's also a problem with the traditional way of monitoring species. And then the last challenge I'll talk about is the invasive nature of some of the methods like the tagging, where you attach a unique identifier to the species. As seen in this diagram, it sometimes causes injury or pain to the species or the organisms, and most at times can also lead to the death of the organisms. And these species are also declining species, so we don't want to cause more death to them. So these are some of the challenges that are associated with the traditional way of monitoring that I would talk about. So scientists came up, or researchers have come up with a novel technique called the environmental DNA. And this is to help solve some of the challenges associated with the traditional way of monitoring or conserving species. So, what is environmental DNA? Environmental DNA is any genetic material that is obtained from the environment without the obvious sign of the source. So, the organism must not be present in order to get its environmental DNA. The environmental DNA can be obtained from soil, it can be obtained from water, sediments, feces, and even the air. We can get the DNA of organisms from the air. Um, as of now, environmental DNA is very useful in species detection and identification, especially for cryptid species, like I talked about. Um, so the general process in environmental DNA is obtaining the sample from the environment. You come back to the lab, you filter them, you extract the DNA, you go through a series of lab processes, and then you identify, you detect and identify the species that was found in the environment. So it's just a simple process. Just like I said, nothing is perfect. So and this is also a new technique that is being introduced. And so there are some limitations associated with the eDNA or environmental DNA application. However, it is much better than the traditional way of conserving species or monitoring species. Now, I've grouped the limitations of eDNA into two categories, the methodological issues and then the ecological issues. So with the methodological issue, there's sometimes contamination at a point of at some point during the protocol. So maybe I go out to collect water samples, contamination may happen or take place, and this may greatly affect my result. And since this is also a novel technique that is being introduced, few studies have been done on some species. And so there are not a lot of reference materials. And also you can get varying experimental results from different researchers. And also with the ecological issues, there's also complexity in the relationship between biotic and abiotic factors. And sometimes there's transport of eDNA from the source environment. So especially when there is heavy rainfall, the eDNA get washed away from the source environment. So you may collect the sample from the environment and end up not getting any DNA from it. So that's also one problem associated with the eDNA application. So this is just a little background about my whole research or study. Now I'm going to go into my species of interest, which is the ringed boghaunter. Now, this ringed boghaunter is one of the only two species under the genus Williamsonia and has been listed as a threatened species here in Maine. The other species under this genus is commonly called the ebony boghaunter. So this is a picture of the ringed boghaunter, and this is a picture of the ebony boghaunter. When these two species, just like I have in my slide, are being put together, it's very easy to tell the differences between the adults of these two species. However, the flight season, that is the emergent season, the habitat preferences, and also when they shed their skin, the exuvia of the ringed boghaunter is very similar to that of the ebony boghaunter. So it's most times difficult to differentiate between these two species, and one is threatened and one is not threatened. So we need to be very careful in trying to conserve these species. Otherwise, you end up wasting, in quotes, resources on the non-threatened species instead of the threatened one. Again, the larvae of both the ringed boghaunter and then the ebony boghaunter is very similar. Although there have been some metrics that have been proposed to distinguish between these two species. However, these metrics are not reliable enough for us to just use physical parameters to tell the differences between these two species. Now, the ringbok hunter has been found in two disjunct regions, the New England and then the Great Lakes, which also include Michigan and Wisconsin. So I have here this graph, which is showing the jurisdictions in which the ebony buck hunter is being found, the hunter is being found, and where both species overlap. So the red shaded region I have down here is showing the jurisdictions or the areas or locations where only the ringed bug hunter is found. And this include Connecticut and New Jersey. And then the green shaded area shows the jurisdiction where only the ebony bug hunter is being found. And this includes some part of Canada and Vermont. And then the purple shaded area is where the two species overlap, where they co-occur. And this include Maine. So care is being taken in order to conserve this threatened species. Um, this dragonfly, just like any other dragonfly, undergoes three phases in its life cycle. The egg stage, then it develops into the nymph or the larvae. It undergoes shedding of the skin, and it becomes an adult dragonfly. And the adult dragonfly spend about 14 days away from its wetland or its location, its habitat, and then it comes back to lay egg on a sphagnum moss, and then the cycle repeats. So why am I even using eDNA in this study? Um, Effort to survey the ringed bug hunter, which is the threatened species, using the traditional method I talked about, have proven challenging. And this is because the emergent season or the flight times of both species co-occur and it's very short. And then also the morphological parameters of the nymph is also very similar to that of the non-threatened ebony boghaunter. So just physically observing and recording data about this species um, will not be accurate, will not be accurate enough to distinguish between the two species in order to conserve the threatened one. So that's why I'm using eDNA in this study. Now, in my research, I asked two questions. One, can eDNA be used to distinguish between the threatened ring boghaunter and then the non-threatened ebony boghaunter? So what I would do here is I would sequence mitogenomes of both species from the tissue samples, and then I would find out the area that makes the difference between the two species from the mitogenomes. And then I would use that to develop and test ring Boghaunter specific primers and probes. And my second question is, can eDNA be a better approach to identifying and then studying the threatened ring Boghaunter here in Maine? So after developing my ring Boghaunter species-specific primers and probes, I would test them on some field samples that I will collect. Okay, so to answer my very first question, um, tissue samples of both the ring Boghaunter and then the ebony Boghaunter were collected from the Maine State Museum by a local expert, Mark Ward, I think he's on the Zoom. And they were sent to us here at the University of Maine. And then since these are tissue samples, I needed to um, break them or lyse them properly in order to extract the DNA from them. So, and they're hard shell. So I incubated them for 24 hours and I extracted the DNA from them using the DNA blood and tissue kits following the manufacturer's instructions. I quantify them to find out how much DNA is in these tissue samples. And then after extraction, part of the extract were Sanger sequence, and then the other parts were Illumina sequence, just to get the full and accurate mitogenomes of these species. So after um, extracting and going through PCR or amplification, the mitogenomes of both species were assembled using a software called the Genius software. And this software was able to tell me the percentage similarities between the two species and also the difference between the two species. And it was found to be 98.6% similar. So genetically, these species are very, very similar. So just using physical parameters or just observing and recording data wouldn't be accurate enough to tell the differences between the two species, especially with the larvae or the nymph. So this is a ring Boghaunter and this is the ebony Boghaunter. The only difference from this mitogenome was the COI region. So the COI region is mostly the region for animal species that will tell the differences between these two species. So I aligned the COI region of both species together again in the Genius software, and I use an inbuilt primer to design my primers and probes. So I align the sequences together. I use an inbuilt primer to design my primer. So this is my forward primer, this is my reverse primer, and this is my probe. To validate my primers and probes, I made sure that the primers were between 18 and 21 base pair long, and they were not self-complementary to either of the sequences. And also made sure that the melting temperature was between 55 and 65 degrees, which is accurate for most primers. And then the Genius software designed the primers for me. And um, I also blasted them on GenBank just to make sure that it does not amplify any other arthropod or any other species. And it did not amplify any arthropod, and which was great, which was what I was looking out for. So now, after designing my primers and probes, I tested them on the tissue samples that I had already extracted from the ringed Boghaunter and then the ebony Boghaunter, and it worked. And I wanted to test for the efficiency of my primer and probe assay. So I did a serial dilution of 6 dilutions from the ringed Boghaunter extracts because the primers and probes that I designed was just for the ringed Boghaunter. So I made a serial dilutions from the ringed Boghaunter extract, and then a standard curve was obtained from series of PCR tests. And I use this equation to determine the efficiency of my primers and probe assay. And it was found to be 92% efficient, which to me, which was it was great. is able to amplify only my ringed Boghaunter, which is what I was looking out for. So that is my first question. I answered my first question. Now to my second question, which is can eDNA be a better approach to identifying and then studying this threatened ring bug hunter? I set out to collect eDNA water samples from various sites in southern Maine. So these 15 sites, some of these sites are known to only the ring bug hunter, some are also known to only the ebony bug hunter, and some are unknown to either of these two species. So those unknown sites, the eDNA method was going to tell me which species was in which wetland or which location. And this is a picture of Magwood. He's been also working on this species, and then he confirmed all these sites that we went out to collect water, eDNA water samples from. So this is a picture of one of my sampling sites, the round pond. And this here is Cindy. She was an undergrad in my lab. She helped me in collecting the water samples from the various sites that we went out to. So what happened at each site was three replicates of the sample of the water was collected into measuring bottles, one from the mouth of the river or the pond, and then we move 10 steps away. We collect another sample, then 10 steps away, we collect another sample. So three replicates were collected from each side um, with globes were changed between each sample just to make sure that you are preventing contamination from happening. And immediately after collecting the water samples, they were placed in an ice cooler just to prevent the DNA from degrading. So after collection of the water samples, they were brought to the Gulf of Maine Research Institute for filtering. So we use the magnetic cup filtering setup. There is there are filter papers beneath these cups. So we pour in our water samples, it drains, it filters, and then we collect our filter papers into tubes, these small tubes here, and we added long Maya buffer just to preserve them and prevent um, the DNA from degrading, since we'll be transporting them back to the University of Maine or running our lab. So now the samples were brought to the lab and we analyzed them. Now, the first thing we did was to extract the DNA from these water samples. We extracted them using the Power Soil Pro kit. The first, the first tissue samples that I extracted, I used the DNeasy blood and tissue because they are tissue samples. But these ones, they can be used for water and then and soil samples. So we use the Power Soil Pro kit following the manufacturer's instructions to extract the DNA from our field water samples. And then the primers and probes that I developed in my question one was tested on these field extracts, was tested on these field samples. However, there were no positive detections from my field water samples. So as seen in this results session, nothing worked, nothing was amplified. So then I went back to think about like why was this not amplifying? Why didn't this work? To rule out the issue of inhibition, I spiked the field samples with extracts of the original ring Boghaunter, what I extracted in my question one. I added I spiked my field samples with the extract of this ring Boghaunter, and there were positive amplification. So this alone is telling me that yes, my primers and probes are working. They are actually amplifying only ringed Boghaunter, which is good, which is what I wanted. Now, as to why there were no positive amplification from the field samples, I was just thinking, and so I came up with some reasons why there were no positive amplification from my field samples. And one reason was prior to me going out to collect our water samples, there were lots of heavy rainfall. So I think the eDNA or the DNA of these species might have been transported away from my sampling site. So that's one reason why I didn't get, or there were no positive amplification. And also, it could be that these are hard tissue species. It could be that they are not shedding enough DNA as we think they are. And then also, which this third reason I am not confident about, but it could also be that they are no longer at these locations that were confirmed. I'm not so sure about that reason, but it's a possible reason why I didn't get any amplification from my field samples. So that was just to answer my second question. So in summary, I asked two questions in this research. One, can eDNA be used to distinguish between the threatened ring boghaunter and then the non-threatened ebony boghaunter? Yes, they can be distinguished genetically, but very slightly. As it was seen, it was 98% similar and just a very slight difference in the COI region of these two species. Um, a qPCR assay, which is the primers and probes, were developed for the ringbok hunter, and it was found to be 92% efficient, amplifying only the ringbok hunter and not any other attribute, which is what I wanted. And my second question, can eDNA be a better approach to identify and studying the threatened ringbok hunter? There were no positive detections from my field samples, and I've shared some reasons why I think there were no positive detections from my field samples. However, future works will be exploring possible reasons why. So for my next steps, I plan on collecting more eDNA water samples, and I want to do this during their emergent season, their flight season. I want to go and collect the water samples during their flight season. With that, at least I'll be sure that I'll get enough DNA from these species. And then I'll come back to the lab and test the primers that I designed on them, do more lab work, PCR sequencing, and hopefully I get positive results and I'll prepare a manuscript for this project or this research that I've done. Yeah. So thank you all so much for listening to my research. I want to thank my advisor, Dr. Erin Gray. I want to thank Mark Ward. He's been very invaluable in this research. I want to thank the Maine Entomology Society for their help, their support, and my other funding sources. Thank you, and I hope to receive questions, feedbacks, and ideas as to what to do to improve. Thank you.
All right, so now we have some time for questions. Um, we have a couple in the chat that I'll start with. So Kathy Claire asked, is the bog hunter endangered everywhere, including the Wisconsin, Michigan populations, or just in Maine?
So in Maine, it is a threatened species. Um, yeah. So in Maine, it's a threatened species. It's endangered in other states. Um, it's yes, it's endangered in other states and also yeah, threatened in other states. But I was just focusing on Maine, which is um it's a threatened species here in Maine. I was just focusing on the main species. But with this diagram or with this map, I was just showing the jurisdictions where these species were also found. But yeah, in Maine, it is a threatened species.
Okay. And also Kathy Claire, did you attempt to identify eDNA of the ebony bog hunter from field samples taken for the ringed bog hunter study?
So because the primers and probes that I designed was just for the rainbow canter, I was is the this assay is just to amplify only ring bug hunter. So even if there's ebony bug hunter, it wouldn't amplify it, so I wouldn't know. I was just looking out for the ring bughaunter and not any other species. So even if there is ebony bughaunter, my primers and probes will not amplify them. It was just to amplify the ring bughaunter. So even if there's any other species, it wouldn't amplify. I did not try any universal primers on them just to find out if there's the possibility of finding ebony bughaunter. It was just my primers and probes that I designed that I tested them on.
Okay. Um, so, Joe, your hand is up. Do you have a question?
Well, yeah, I do. Let me lower my hand. Okay, Christina, thank you so much. Wonderful presentation. And I want to concur with you and with so many in the field that there is a taxonomic crisis in terms of that skill set as we're moving on, and sort of reiterate that eDNA augments that skill, the field skill of being able to go out and identify with your eyes or with a camera or binoculars, um, as well as other techniques that may be useful. But nothing's ever going to replace, contrary to what people outside of the field may suggest, that we could just use DNA. We don't need taxonomists or people getting PhDs in this stuff. We absolutely do. We need that informed knowledge base to certainly know where to start to look, to understand habitat as well. And that gets me to the first observation slash question, and that is also noting eDNA and molecular work is sort of a black hole in my own background. I do chemical ecology and general ecology. I tend to play in everybody's sandbox, but not this one. So bear with me. But it seems to me that one of the things that really would enhance the skill of using eDNA like this as an entomologist slash ecologist to sort of look for organisms would be to have a really good understanding of the watershed. And I want to cite your reference to the fact that it had rained a lot, and that may be one of the reasons why you didn't see that DNA. And so as you were saying that, I'm like, well, that water had to go someplace. It could have evaporated, depending on the type of pond, if it's a kettle pond or something. But if it has an outflow, and we understand hydrology in that area, then we might be able to focus our efforts, even in drier periods, to where that outflow is occurring and sample from the moss or the soil in those areas. And so um, uh, I wonder if you could comment on what you think about enhancing our effort on the ground in that manner.
Yeah, so with my research, I just collected water samples from the actual site. So
Yep.
I didn't look at any other area, any other environment. It was just collecting water sites from. If this is the pond, this is the pond I'm collecting water samples from. I didn't like look out for any other. So I think, yeah, your idea is also something that we can look at, since we can also get DNA from the feces, from the soil and all that. It's something we can also look at as researchers or as scientists. But yeah, I didn't look at any other. I was just collecting water samples from my site.
Yeah, that's kind of where I think it's I'm interested in this DNA business, and I was just talking to somebody about it recently. I'm really eager for eDNA to become a black box that I can, like Star Trek, just do my tricorder scanner. And some of you really appreciate that reference. Um, and I don't want to be a molecular biologist. I have no more bandwidth, and as I'm getting older, not enough brain cells. But I do think having a fundamental understanding for how water would move in an area. And as you're speaking, I don't know the pond that you're talking about, but I'm going back to a childhood pond or bog, Dresden bog, in mid coast area, and I know that it was a dammed bog, so it turned into kind of a lake, and it has an outflow. And I'm just envisioning that there'll be a period when there's not much outflow coming out, yet all of that water was sort of going in that direction. And I could have gone, I couldn't have, I wasn't doing this work, but if I were sampling there, I would go to that spot if it looked like I was missing things all around. I'm just wondering if it's a natural concentration for eDNA. And so as I'm speaking, and ideas are cheap, of course, I'm wondering it would be interesting to do like a survey from the upland down to the lowland to see if we start to see eDNA from any particular organism. You could use a common control organism, then rare ones, as you were looking at, to see if it starts to increase in some way so that it's easier to find. And I think that would enhance our field techniques regarding eDNA because I still think we're in the early stages.
Yeah, for
Yeah.
sure. Yeah. It's something I can consider during my second round of sampling. So I intend
Well,
some.
you're doing a whole lot of work right now, and I certainly don't think that you need to change the world. I'm just sort of speaking out loud, and
Yeah.
you've got a
Yeah.
lot more time left in it than I do, so maybe you can influence this as you move forward.
Yeah. Yeah. I mean, it's something I can consider looking at the fact that I'll go out again to collect water samples. So yeah, it's something we can look at.
Cool. That's
Okay.
all I have.
Thank you.
Does anybody else have a question? I know we have one in the chat. But does anyone have anything they want to unmute themselves to ask? Uh, oh my God, I'm missing it. Oh, so Trish in the chat asked, how old were DNA samples taken from the archives? Or I assume that means the museum collection. And is it possible that the DNA sequencing has changed for the ring species over several generations due to environmental pressures?
So how old? I think I'm going to leave that to Mark Ward. He collected the tissue samples from the museum for me, but I don't think the sequences have changed. Um, few works have been done on this species in terms of its sequences. So the sequence that I obtained from my Sanger sequencing and Illumina sequencing, I think was the first one. So I'm here to post or share that on GenBank. I didn't find any reference sequences in order to even compare. So with that, I don't think the sequences have changed. Yeah, if that answers your question. But with the age, I think I'm going to leave that to Mark Ward. He's on the Zoom. He can help me with the age.
Those samples were definitely collected in certainly within the last 20 years. And I think most of them were much more recent than that. So just like in the last few years. So
Does the DNA remain stable enough to extract from museum specimens like dried specimens? Instead of because I know we talk about having samples in, minus 80 C freezers
Yeah,
to keep them stable. But like
yeah.
dried museum specimens can still be used even if they're 20 years old or so.
Yeah, they can be used. If you're able to preserve them well enough, you can use them. You can get enough DNA from them because it took me a while before extracting the DNA from the tissue samples. So they were in the minus 80 freezer for a long time. And I still had DNA, just that, yeah, I still had DNA and I was able to amplify and get the sequences out of them. So I think if you're able to preserve them very well, you should. You wouldn't have any problem with DNA degradation. So
And we have another one. Do you think different times of year might have different sampling results? For instance, is there a time of year when nymphs would be more numerous in the water and therefore might offer more DNA that could be picked up? And I kind of had a similar one I was thinking of, like different times a year might have different proportions of later instar larvae that might be bigger and might be pooping a lot more than smaller instar larvae, something like that.
yes, I think so. The last time I went out to collect samples, I think I did that in mid-July. Yeah, mid-July and somewhere in October. So for now, and then their emergence season is from April to May. So this time around, I want to go out and collect during that time and just see. So hopefully I'll get to know um, if the seasons is affecting the DNA being shed in the environment. But I think, yeah, it has an effect on them. It has an effect on the DNA. So,
And another one from Hillary. Kind of an out there idea, but how often are the larvae eaten by fish? And are there fish species that you could possibly sample that got contents?
ooh, that's a good one. For that, I cannot tell, but it would be a cool idea hmm, to also sample.
Because I know similar things occur with like stable isotope testing, things like that. Wonder if those similar methods could be applied.
Huh.
Can I say something here? Most of the sites, the sites where this species is found, the aquatic sites are sort of more, they tend to be fishless
Yeah.
sites. All the sites that you sampled were all fishless sites. They're sort of more like long-term vernal pool type
Yeah.
habitats. They can be found in places that also have some fish associated with water bodies, but none of the sites that Christiana worked at were of that ilk. So they were all fish-free environments.
Yeah.
All right. Does anybody else have any more questions? Yep, just one more. How effective do you think sediment sampling would work for the nymph stage?
Hmm. It for I cannot tell unless I go out to collect sediment samples to find out. But hmm, sediment samples and water samples, I feel like water samples should get me more DNA than the sediment samples. Um, the eggs are always being laid on the surface of the water, and so getting sediments, I might get DNA, but I feel like from water samples, getting DNA from water samples will be better than getting them from the sediment samples. That's what I think.
Okay. And what about doing some kicking around of the sediment a lot before sampling? Although maybe
Sorry.
you've already. What about kicking around the sediment a lot, like kicking it up and then sampling from there?
Hmm.
Kind of like
I-
kick netting for aquatic inverts with a D net, you know.
Hmm. It's I would say it's all things we can I can talk about with my advisor or consider when going out to collect my another round of sampling. So yeah, I feel like it will be interesting, but do I have all the time? I don't know. But yeah, I would try. I would consider all of these ideas and see. Yeah.
And Bob, do you have a question?
Yes, I was just thinking if you actually were able, you wouldn't want to kick up the sediment because that would mix material from multiple layers. Your sphagnum bogs generally are going to be way too acidic for any fish, but your sediment record preserves an incredible history of the basin. If you can find any in the surface sediments, which is going to include degraded material from all the organics that were left down there, you could potentially do a core of the sediments and look at changes over time.
Mm. Okay.
Of course, this is way beyond what you're looking at, but
Yeah.
kudos. That was a fascinating presentation. Kind of a bummer you didn't find anything, but
Yeah.
congratulations, and takes guts to say, I'm going to find out why.
Yes.
as opposed to just throwing your hands up. Okay, that didn't work. I'll go on to do something else.
Yeah.
Are you sampling again this summer? You're doing a
Yeah.
round two.
Yeah. I want to do that during April, May, thereabout. Yeah.
Cool. Well, does anybody else have any more questions? Hillary? All right. Well, if nobody else has any more questions, thank you again, Christiana. That was a great talk. It's fascinating. Um, and so everybody feel free to join us again in a couple weeks. I believe February 5th is our next webinar. Um, I don't have it in front of me, but I think that's Colleen Tierling giving a talk about biocontrol on woolly adelgid. So join us for that one, 7 p.m. on a Thursday evening. What better time to talk and think about bugs as we inch closer towards bug season.
Yeah.
So thank you very much, everybody. Have a wonderful night, and thank you again, Christiana.
Welcome.